Hey everyone, we're going to do the next part here of biotechnology, and I want to talk about two processes with you guys. I want to talk about this thing called PCR, which is probably one of the most commonly used lab techniques in molecular biology labs today. And then this other thing called CRISPR, which is actually kind of new. I would recommend that you watch the video I have linked on CRISPR. It's a really good resource, and it takes you through some animations that show it to you. I'm just going to talk to you guys a little bit about it so you have those extra notes. So I want to talk to you guys about PCR first. PCR is an acronym. It stands for Polymerase Chain Reaction. And the easiest way to think about this is it's kind of like a DNA photocopier. So PCR is used to make lots and lots of copies of one small piece of DNA. It uses enzymes that we've talked about before. So it uses this enzyme called DNA polymerase. But the DNA polymerase is actually derived from a very special organism. It's derived from an archaean that lives in hot springs. So this enzyme can actually be heated almost to boiling, and it doesn't denature because of how that enzyme evolved. So that means we can actually use heat to separate the DNA strands rather than having to use enzymes to separate the DNA strands. And this leads us to how PCR works. So PCR is actually done in this instrument, and there's a little cartoon of it here on the slide, called a thermocycler. This is basically a heater. You can heat it up and cool it down very, very quickly. So what we do is in a test tube, very tiny test tube, we put the DNA that we want to copy. We put in primers. So these are pieces of DNA that will stick to our DNA so that DNA polymerase can copy. But we're going to build those primers so that they actually attach to very specific sequences. We're not going to copy all of the DNA, we're only going to copy short segments. And so the primers will allow us to target which pieces of the DNA we're actually going to copy. We'll also put in the tube nucleotides, because we need those as raw material to make DNA. And then we'll put in this thermostable polymerase. There's also some other things in the mixture, but those are the important pieces. And then what we'll do is we're going to heat the DNA up to 95 degrees centigrade. And you can see here what happens is the DNA strands separate. So we're doing this instead of using DNA helicase. We then cool the DNA down to about 55 degrees centigrade, and at that temperature, the primers will stick to the DNA, or they do this thing called annealing. And then we'll warm the samples up, the tubes up to 72 degrees centigrade, and at that point, the DNA polymerase becomes activated, and it will do the copying. So when we do this once, we end up with 2 copies. When we do it twice, we have 4. By the time we've done it 3 times, we have 8. So this thing grows exponentially. So what that means is by the time I do this 35 times, and that's usually how many cycles that we run in this, you end up with over 1 billion copies of the DNA. And that's just assuming we start with 1 copy. Now here's the advantage of doing this. If you have very small samples of DNA, it allows you to make many, many copies. So we use this a lot in forensics. If you've ever done 23andMe, they use PCR to amplify certain sections of your DNA so that we can analyze the gene sequences. So PCR is used quite a lot. Now, I know this is kind of ironic, but I'm going to actually switch over here to show you something on YouTube. I'm going to show you this video on YouTube that actually takes you through what's happening here. So I'm going to put this in full screen. And you can see here's the molecule of DNA. Now it's in the test tube and we're heating it up. When we heat it up to 95 degrees centigrade, we're going to break the hydrogen bonds between the nucleotides. Then we'll cool the sample down to 55. And at that temperature, these primers that we've added to the tube are going to anneal or stick to the DNA. And notice they're sticking in very specific spots. Then we're going to warm the sample up to 72 degrees centigrade. And at that point, my DNA polymerase, it's called Taq polymerase, will attach and become activated. And we're going to copy the DNA, so we're going to make double-stranded molecules. Now we've only really copied this one little section that we're interested in. So that's cycle 1. Cycle 2 will repeat. We'll heat, We'll cool, we'll allow the primers to anneal, we'll warm it back up, the DNA will be copied by the polymerase, and we heat it up again. So now we're in cycle 3. Now when we complete cycle 3, so we attach the primers, now we end up here. These are our target sequences. So those are the pieces of DNA that we actually wanted to copy. So this process, we're going to continue it. We're going to do it a bunch more times. And every time we do it, we get more copies of our DNA molecule. So that's the idea behind doing PCR. We'll talk a little bit more later about how we can use that. Now, another technique that's actually very, very new, and again, there's a video I have on CRISPR I really like. I would highly recommend that you watch it. But I wanted to talk about what CRISPR was. So CRISPR is another acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's the CRISPR part. Now there's this other piece called Cas9. Cas9 is actually a protein. And CRISPR-Cas9 was originally derived, again, from bacteria. A lot of stuff from bacteria. CRISPR-Cas9 acts like an immune system for the bacteria. So here's how CRISPR works in bacteria. What happens is if a bacterial cell gets infected by a bacteriophage, that virus is going to insert its DNA into the host cell, and if it's successful, it's going to take over the genes of the bacterial cell, turn it into a viral factory, and kill the bacteria. So bacteria don't sit around waiting for this to happen. Over evolution, they've evolved this system called the Cas system, and it's this Cas protein will go and take foreign DNA and actually break it down. So it breaks down the DNA and then inserts little pieces of this DNA into part of its own DNA. This is what's known as a CRISPR array. Now, this is then used when the DNA gets transcribed to build another protein that's similar to Cas. It's called Cas3, and that can then target viral DNA. So if the cell gets infected again, it can target the viral DNA and break it down. What scientists realized is they could actually use this system to modify DNA. So what CRISPR does is we can modify it in the lab with different target systems. So this kind of yellowish thing, this is the Cas9 CRISPR system. So there's a little piece of nucleic acid in there, and what it'll do is it'll go in to DNA in a host cell, and it'll cut and edit that DNA. So we literally have the ability now to edit DNA in a living cell, and we can modify that DNA while the cell is still alive. So we don't have to insert the DNA like we did when we had a vector, and we can make changes to a cell as it is.